Diabetes, biotechnology, and human health. The potential to cure human illnesses and improve general health with biotechnology is immense, and many applications of biotechnology are already in use. Sequencing the genomes of tumors to identify their mutations and treating patients with targeted treatments based on the findings. Using cancer patients' own T cells to fight cancer by stimulating them to increase their number in the lab and then returning them to the patient's blood. Harnessing genetically modified bacteria to clean up toxins and other pollutants in the environment. Developing safe, highly effective vaccines to prevent infections by pathogenic microbes. Treating human diseases with proteins made by microorganisms. Diseases that are caused by one mutant gene or one missing protein are excellent candidates for biotechnology cures. If scientists can repair that gene or replace its protein in the right place and time, the disease is treatable. Multi-factor diseases are harder to understand and treat. Diabetes is caused by the low level of one protein in the bloodstream that prevents the proper production of insulin. By using biotechnology to increase the level of this protein in the bloodstream, diabetes can be successfully managed. In order to develop this treatment, scientists need to use knowledge about genetics, molecular biology, microorganisms, and medicine. From the Centers for Disease Control and Prevention on Diabetes. If you have type 1 diabetes, your pancreas doesn't make insulin or makes very little insulin. Insulin is a hormone that helps blood sugar enter the cells in your body where it can be used for energy. Without insulin, blood sugar can't get into cells and builds up in the bloodstream. Cells are starved of the energy they need. High blood sugar is damaging to the body and causes many of the symptoms and complications of diabetes. Diabetes affects approximately 200 million people worldwide. This number is predicted to rise at a continued exponential rate. The level of glucose in a person's bloodstream is measured by sampling a small amount. A person with diabetes lacks the ability to effectively control the level of glucose in the bloodstream. This results in many problems, including heart disease, kidney failure, and blindness. Type 1 diabetes is usually diagnosed at a young age. Its cause is primarily genetic, unlike type 2 diabetes, which is a disease of insulin resistance that likely develops from a combination of both genetic or environmental factors over time. Type 2 diabetics make some insulin, but not enough to keep their blood sugar at a healthy level. It was discovered in 1921 that many people with diabetes lacked the proper amount of a protein hormone called insulin. At first, doctors treated this ailment by injecting insulin harvested from cows, bovine, but this cow's protein is not the same as human insulin. The human immune system began to reject the cow hormone substitute and caused complications for the patients. In the late 1970s, scientists began to look for a way to make human insulin in a laboratory. This resulted in the first product ever produced by recombinant DNA technology, a specific type of genetic engineering. In this case, the gene for human insulin was found and isolated. It was then put into the bacterial chromosome of E. coli. The bacteria then was able to produce human insulin and could produce it very rapidly. The hormone now known as humulin could be harvested. The protein can be extracted from bacterial cultures using various techniques. It can then be delivered to the patient. Currently, there is no cure for diabetes, but with advancements in insulin therapy, patients can now avoid many of the life-threatening complications. The DNA Learning Center has created a slideshow explaining how human insulin is made using bacteria. You can click the link here to watch the video and animation, but we're going to summarize it in the next few slides. Knowledge of the desired protein is necessary to address the problem. Insulin's amino acid sequence was determined. Insulin is a small peptide hormone of two peptides linked together by two disulfide bridges. The circles are the amino acids. So here's the primary structure of the insulin protein. We have two peptides, the A chain and the B chain, linked together by these disulfide bridges. For each peptide, A and B, a DNA sequence was synthesized that could encode it. That's that first step of DNA sequencing that we talked about in the last section. Programmable DNA synthesizing machines are used in many labs, which is followed by electrophoresis. DNA polymerase was used to make the second complementary strand of each DNA molecule and to make many double-stranded copies of it via PCR. The double-stranded DNA encoding the peptide was inserted into plasma vectors via bacterial transformation. Bacterial colonies carrying the gene for insulin A chain or B chain are expanded by growing them in large liquid cultures. The bacterial cells were induced to express the insulin genes, and they released the insulin protein into the liquid culture media. The two chains of insulin peptides produced in the bacteria are mixed together and then purified. The next step is to combine the two chains by forming the disulfide bonds. Finally, the insulin is ready for injection. So in this large liquid medium, we have bacteria producing insulin chain A, and in another, we have bacteria producing insulin chain B. We have to purify the insulin peptides, then combine them so that disulfide bonds form, and then the insulin is ready for injection. Because insulin is a small dipeptide, it spontaneously folds into a shape that is recognized by human insulin receptors. It is ready for injection. Since insulin is a small peptide hormone, it is an excellent candidate for production by bacteria. Larger proteins usually do not fold correctly or get modified correctly in bacteria because bacteria lack endoplasmic reticulum, Golgi apparatus, and the proteins that help with folding. Recent advances in biotechnology allow us to produce more therapeutic proteins in cultivated mammalian cells so that they are folded and modified correctly. Mammalian cells grow much more slowly than bacterial cells and reach lower cell densities, so using mammalian cells is more difficult, expensive, and time-consuming. Medicine is being revolutionized by more and more targeted, genetically engineered therapies for diseases. In addition to diabetes, many types of cancer, autoimmune diseases, and some types of heart disease can be treated with genetically engineered proteins. The discovery of plasmids and enzymes that modify DNA in the 1970s began a 50-year revolution in molecular biology, agriculture, and medicine that continues to this day. Diseases that are caused by one mutant gene or one missing protein are excellent candidates for biotechnology cures. If scientists can repair that gene or replace its protein in the right place and time, the disease is treatable. Multi-factor diseases are harder to understand and treat. Diabetes is caused by the low level of one protein in the bloodstream that prevents the proper production of insulin. By using biotechnology to increase the level of this protein in the bloodstream, diabetes can be successfully managed. In order to develop this treatment, scientists need to use knowledge about genetics, molecular biology, microorganisms, and medicine.